Now that you've identified one or more modules of interest using tool 1 or 2, it's time to examine these modules in more detail. In doing so, you may learn more about biological processes that are active in your experiment or processes associated with your genes of interest. It's unlikely that a typical gene module can be described completely using a single semantic annotation due to the complexity of gene regulatory networks. That's why we created an annotation page for each module that contains a variety of information about that module. So let's take a look at what we can learn about each module by using tool 3. For example, by using tool 2, I had determined that my gene of interest, say 48, belongs to module 185. So I go to the annotation page for module 185. The first thing I see is a plot of module activities calculated for each of the 716 experimental contrasts derived from a diverse compendium described in the paper. Those in which this module explains more than 5% of total gene expression variants are colored in yellow and blue. The first table below shows ranked contrasts that give a positive SVE value, and the table below it shows ranked contrasts that give a negative SVE value. Remember that what you care about at this point is the absolute value of SVE. And the contrast that produces the highest absolute value of SVE for this module is this contrast here. So what this means is that module 185 explains 19% of total gene expression variants in this experiment. It's not super clear to me by reading these descriptions what the two conditions being compared are. So I can go to the GEO page that describes the microarray experiment from which this contrast was derived and read the descriptions of all of the conditions. And by doing that, I learned that the two conditions being compared here are worms exposed to light for 6 hours versus worms kept in the dark. Note that the order of condition 1 versus condition 2 is important to keep straight. If these two were switched, then the SVE would have the opposite sign because all of the fold changes would have the opposite sign too. The same type of analysis has been performed separately for the upregulated and the downregulated genes in each of the 716 contrasts. This can increase the sensitivity of module detection. For example, I see that genes upregulated by DAF19 mutation strongly activate these modules, and by looking at the downregulated genes only results, I learned that it's the downregulated genes in this light versus dark experiment that strongly activate this module. So to summarize, these contrast SVE results told me that the module that my gene is part of is activated by genes that are downregulated in response to light and by genes that are upregulated in the DAF19 mutant. As I scroll further down, I find GO-term enrichment results listed separately for each Hemi module. Remember that Hemi modules are genes that are regulated in opposite direction within a module, with A being the main Hemi module, that is, it contains more genes. My gene, say 48, was part of the A Hemi module, and I see that this A Hemi module is enriched for GO terms related to non-motile cilia. This is also a good example to show that genes in the two Hemi modules can have very different annotations associated with them. Next down is the anatomy or tissue enrichment. These were calculated by testing enrichment of module genes within genes that have a confirmed expression in specific tissues, as reported on WormBase. In total, over 2,000 anatomical terms were interrogated. Here I see that genes in Hemi module A are enriched for genes with known expression in ciliated neurons. Next, I find transcription factors, known transcription factors that have overrepresented motifs within promoters of genes in Hemi module A. I note UNC86 here, which is known to be active in neurons. Also, it's interesting that SAE48 itself is here on the list. However, it's kind of low on the list, so it could be a false positive, and the false positive are designated by these red asterisks. It's important to note that Homer only looks for these 108 transcription factor motifs, which you can look at here. So if you don't see a result for a transcription factor of interest, it could be that Homer just doesn't look for it. So next down is another method, and so the same for hemimodule B. And typically there aren't as many results for hemimodule B because it's the smaller hemimodule. So here is another method for identifying overrepresented sequences within gene promoters called Moby Dick. And these words provide another opportunity to identify regulatory factors in the module, especially if a transcription factor of interest to you is not one of the 108 that are known to Homer. Moby Dick was also applied to 3' UTR sequences because 3' UTRs contain important regulatory elements, including microRNA and RNA binding protein sites. None were found to be enriched in this particular module. And finally, all the genes that are part of this module are listed at the bottom of the page with their links to Warmbase. You can use this information to do your own analyses of the module, for example, by cross-referencing the module genes to gene sets of special interest. To summarize, by using this annotation page, I found that on the whole, genes that covary with SAE48 are repressed by exposure to light and are upregulated in DAF19 mutants. They're enriched for annotations that have to do with cilia and are expressed in ciliated neurons and are potentially regulated by UNC86 and maybe even autoregulated by SAE48 itself. This information is consistent with known neuronal functions of SAE48 and can also be used to generate further hypotheses about its function and regulation. For a table that summarizes annotations of all modules and suggests a consensus process that the module represents, please see file S2 associated with the manuscript, which can be accessed here through Figshare. So it is this guy here, file S2. You can download it, open it, and that's what it looks like.